Alrighty. Hello, everyone. I'm sure you have really missed the sound of my voice, right? Teach AP Bio online, they said. It'll be fun, they said, right? No one said that, but we'll get through this together. I wanted to take the time to go over the stickleback fish activity that you guys worked on over the past couple days here. I do have the answer key for this posted just as a document, so you can go over it that way, but I wanted to kind of go through and explain everything just like I would in class, so that if you had anything that was unclear or you weren't sure about why an answer was an answer, I could talk it through. And then of course you can always email me, comment on this video, whatever you want to do, reach out somehow so that we can get things cleared up, because at this point it looks like College Board is still planning to administer exams. Not sure about that, but we're going to keep on plowing ahead with that idea in our minds. All right, so you were following up with from the video we watched together in class about the stickleback fish and their switches, or what we're calling enhancers. This is all about having this PITX1 gene, which is a gene that is in all stickleback fish, but the way that that gene is regulated will dictate what kind of traits these fish get. So this is just the cover page here. We will scroll down to the first page where you had to fill in, this diagram. So you were asked to label the diagram with a bunch of terms that were given to you. The first one, the protein coding region, that's going to be this area of the DNA. This is the actual gene that's going to be transcribed. That would be the PITX1 gene. The regulatory switches, or what our book calls enhancers, are these areas that are upstream from the gene, that rhymes, where you can have different things or different parts of the body that would be expressed depending on what these switches are. C, the promoter, that's the beginning area, right, of the gene where the RNA polymerase binds to get that party started. And then D was your mRNA, and you would know that because it is our single-stranded molecule here, and then our RNA polymerase, the enzyme that does all the work here in transcription. All right, so when we're talking about the stickleback fish, we're focusing in on the transcription part of eukaryotic gene regulation. Remember, there's that methylation, acetylation, that's all at the DNA level. What we're looking at here now is how the mRNA, or the transcription part of protein synthesis, is being manipulated or regulated. All right, and you were asked to describe the function of regulatory switches. So you should have said something about how these switches bind to specific transcription factors called activators or repressors, and they deal in turning on these different types of tissues in these fish. And then you were asked for number 3 to circle words that are involved in eukaryotic gene expression and or regulation. So proteasomes, definitely not part of the process. Activators, yes, those are those specific transcription factors that can turn a gene on. DNA polymerase, nope, wrong process. General transcription factors, absolutely. Those are those things that are going to combine with the RNA polymerase to create that the transcription initiation complex, so you should have circled RNA polymerase as well. Mediators are another name for specific transcription factors, so you should have circled them. Operons, remember, those are our bacteria, our prokaryotes. They're doing gene regulation with operons, so not eukaryotic cells like a fish. Eukaryotic organisms like a fish. Lactase, that's an enzyme, not involved here, although it is involved in the other assignment you guys are working on right now. And because we're talking about transcription, no ribosome in this part of the story. All right, so you shouldn't have circled that one either. Okay, so then moving on here, which protein from the list above binds to the regulatory switches? You should have said activators for that. And then which proteins from the list above bring the bound activators in contact with the proteins bound to the promoter? Mediators for that one. I'm thinking you could also say DNA bending protein, but that's not listed here as an option. So we're going with mediators, and mediators are just specific transcription factors. All right, so then you were asked to draw in all these components that you were given. So, sorry, I have Moby written next to it because usually I draw this out with my Moby, but, different times. Back in 2019, before the virus. Anyway, these areas are your regulatory switches or your enhancers. We can call them same thing. You also have your activators. These are the proteins that bind, or the specific transcription factors that bind to the enhancers. Mediator proteins, these are just transcription factors, is another name for them. More transcription factors. The promoter is that area in front of the protein coding region, and then our RNA polymerase. All right, so your picture should look something like that. All right. And then moving on. Sorry, my dog's snoring really loud. I hope you can't hear her. Some questions to answer. Number 1, list all the tissues shown in figure 2 that express the PIDX1 gene. So you should have just said pituitary, jaw, and pelvis. You were leaving eye out of this one because if you did the reading, it mentioned how these fish, or the PIDX1 gene, is not transcribed in the eyes because these fish don't have a sequence that can bind to the activators that are present in the eyes, so you don't have that happening. All right, number 2, if a fish does not produce activator 1 proteins because of a mutation in the gene that encodes for proteins, PIDX1 will not be expressed in which of the following? So because this is activator 1, sorry. So because activator 1 dealt with the jaw, you should have not crossed off the jaw in this case or checked off the jaw. We're leaving the eye blank because we're told there's no switch for the eye, nothing to be expressed in those cells. So that's off, so you should have just checked off pelvis and pituitary. Number 3, if a fish does not produce activator 3 protein, so the 3 one was for the eye, if you look at the picture that's on the previous page here. So again, you would be leaving the eye unchecked. You could still get expression of this gene in either of these 3 parts of the body, the jaw, pelvis, or pituitary, which is a gland. All right. Number 4, assume a fish inherits a deletion mutation in the pituitary switch, which inactivates that switch. Draw an X over the mutated switch in the appropriate tissue. So you should have drawn an X in all of the switches, or all the pictures, even though the eye, that part of their body, this gene is not expressed. The DNA is going to be the same in every single one of these cells. So in the pelvic cells, in the pituitary cells, cells of the jaw, all the parts of the body there, that same mutation would be inherited. So that same mutation would be in all the DNA, all of those cells. So you should have put an X over all those switches. Okay. Number 5, when a mutation in the pituitary switch prevents activator 4 from binding, where would you expect PIDX1 to be expressed? Activator 4 was the pituitary activator, so that would have bound to here. If we don't have that activator, therefore we're not going to express this gene in that particular part of their body, so you should have just checked off jaw and pelvis for that one. Okay. Number 6, a fish inherits a mutation that results in a new regulatory switch, an eye switch. Ooh, fancy. So now you should have checked off all of the choices here because of this new switch, and we have the activators for it, we're able to express this gene in the eye. Can't imagine what that would look like, but for the sake of the question, you should have all 4 boxes checked off. Number 7, a fish inherits a mutation in the PIDX1 coding region. Draw an X over the PIDX1 coding region in the tissues where you would expect to see the mutation. So because this is a mutation in the gene itself, it tells you it's a nonsense mutation. If you guys remember, that is a substitution mutation that results in a premature stop codon or a really short and non-functional gene. That's what truncated means, short. So in this case, again, if the mutation itself is in the gene, then all of these cells are going to have the same mutation. All right, all of your DNA, remember from our discussion when we were all last together, is the same in all of your cells. Okay, and I have a little note over here too to help you. Pretty good so far? Hope so. All right. Where would you expect PIDX1 to be expressed? Well, you shouldn't have checked off any of them because if this is a case now where you have a mutation in that gene and the protein that would come from this gene is going to be affected or not made, truncated, non-functional, you would not be able to express that protein in any of these parts of the body. Okay. Number 8, you were basically just picking the fish that doesn't have a pelvic phenotype or a pelvic stickle. So you were picking B for that one. Easy enough. And then number 9, you were asked about, or you were given a scenario with stickleback fish and their fossils. We were starting to watch that in the video on Thursday, but we ran out of time, and I thought we could finish the video on Monday, but that didn't happen. Hopefully you were able to review it on your own time. But anyway, you were told that these fish lack a pelvis, so you should have selected the pelvic switch crossed out. That was part C. If this switch is not working, you're not going to be able to bring this part of the DNA close to this gene to have that particular protein expressed in the pelvic region of the fish. All right, and it would yield a fish that looked like the fossil with no pelvic stickle. All right, so that is our stickleback fish review. Again, if you have any questions about this stuff, please feel free to reach out, post a comment, email me, whatever you prefer to do. If you're not sure about something that I discussed, you're not sure about an answer, I would be happy to help you. Keep up all the good work. You'll be likely hearing my voice more in the coming days here. Thank you, everyone.